This method can answer key questions about the phosphorylation of the 5' end of DNA or RNA molecules by a special class of enzymes known as polynucleotide kinases.
The main advantage of this technique is it has the resolution to detect very small changes in DNA and RNA substrates.
Begin by preparing the RNA enzyme kinase reactions. For each reaction, combine 1 microliter of 500 nanomolar RNA substrate, 8.3 microliters of 130 nanomolar LAS1 GRC3, and 0.2 microliters of 5 millimolar EDTA. Set the heat block to 37 degrees Celsius, then mix 0.5 microliters of an ATP substock from the concentration series with 1 RNA enzyme mixture, and place the reaction on the heat block. Continue mixing the reactions and placing them on the heat block at 10-second intervals. After a 60-minute incubation on the heat block, quench each reaction by spiking it with 10 microliters of urea loading dye. Immediately perform downstream analysis or store the reactions at -20 degrees Celsius to be analyzed at a later date. To prepare a 15% denaturing acrylamide gel solution, combine 22.5 milliliters of premixed 40% 29:1 acrylamide bisacrylamide solution, 6 milliliters of 10x TBE, 28.8 grams of urea, and RNase-free water to a total volume of 59 milliliters. Then, gently stir the solution. Heat the solution in the microwave for 20 seconds. Stir it, and immediately return it to the microwave.